Alright, and so once it's finished, you can actually go and take a look at all your data. There will be a couple of videos, it should just be two. The first one is your phase images with the fluorescence overlaid and the numbering or contouring if you did that. And the second one is just a running graph of the fluorescence for the whole cell fluorescence or the ROI fluorescence if you did an ROI. In the CSV folder, there will be a CSV file with the whole image and region of interest fluorescence by frame. In the main folder, when you run the tracking, it outputs lineage data.csv as well as global cell statistics.csv. So lineage data is just your individual lineages, as I showed you before, and the doubling time and that kind of thing. And the global cell statistics is the time, cell count, and the mean fluorescence for the just cells, not the whole image, but just cells. And this looks like the raw value. And then if we go to where we output the lineage file, you can see here you got frame, time, area, XY position of the cell, and fluorescence if that was available. If you want to go and output more of these files, say you realize you want to look at all of them or something, or say you want to remake the videos maybe without the lineage tracking numbers written over them, or if you want to rerun the tracking and this time track every 5 images instead of every 15 images, you can do that and you don't have to go through the entire process again, which is super nice. Instead, you can just run the script and answer no for things you don't want to do. So we already have aligned images, and we don't need to train a classifier. We've already tracked the cells. I do want to output more files, so I'm going to say yes. And I already have the video, so I'm going to say no there. And it's going to ask me, we want to write aligned, because that's where our images actually are. It's going to ask me if I want to render the videos, and I'm just going to say no, I don't want to re-render videos. You can enter the name of the directory to output the file to, so CSVs. And do I want to output all of them? Let's go ahead and say yes this time, and I'll go ahead and output all of them. You can see it's an extremely quick process. I now have 1,400 files. You can go ahead and take a look at all of the files. Very useful. Or write another script to look through them for you. That's why Python is neat. So that's how that works. Now I'm going to go into a slightly more advanced users section. So just in case you want to maybe re-render the videos or do some other stuff, you can always go and actually edit the scripts. So say I look at this video and my fluorescence is insane. The amount of green just goes and spreads all over the video, that kind of thing. I can go into imageanalysis.py, open it with whatever your favorite text editor is, and you can see here there's a lot of comments. It's a lot to look at, but all of the parameters for video rendering are right here in this IFB render section. Super useful, and you can go and actually change the different channels. Right now there are 4 channels because theoretically this could run up to 4 different channels, the first being phase and then 3 fluorescence channels. We only ever right now have them set up for 2. If you wanted to set them up for more, you can go through and edit that and do that. Right now we only have them set up for 2, and so you can actually change these numbers here. Blue, green, and red, so this number here is affecting the actual color. If these are all even, it's going to be like black and white. Or you can make it more green or more red. So say if I didn't have GPP, maybe I'm using it cherry, I could go and make it red instead of green. We have the shift here. So this is just deciding how much to shift. Right now I have it set where if there's no fluorescence whatsoever, it doesn't shift it, but if there is fluorescence, it does shift it to allow that fluorescence to come out. And we also scale it, and I have notes here telling you how the shifting and scaling works. And if you just go ahead and save that, you can go and run this again, and this time I just answer no, no, one, yes, 448, 45, 449, 10, 2017, 412. And that's it, we're in cherry. And do I have masks? You can sell it, no, you don't actually need the masks. You just use them to outline the cells, and I don't feel like doing that right now. No, no. And if you go ahead, it'll run its course. And if you go look at the video now, it's red instead of green. So these values here are how you change that, in case you were wondering or you're interested. And there's a lot of other things you can do with these scripts, so feel free to explore, change them as you desire, write some new ones, and have fun with it, basically. So that was Cell Tracking with me, your host, Heather Dieter. And please go actually read the book chapter and take a look at that, because that'll answer, I'm sure, a lot of the questions I might not have gotten to here. Thanks, guys. Bye.